Hello students, in today's practical we will perform sequence alignment using BLAST software, that is basic local alignment search tool. So generally in BLAST program it will find a region of local similarity between sequences. So there are different types of BLAST program, but here we will perform nucleotide BLAST for a given nucleotide sequence. For aligning any sequence through BLAST, we first have to go to BLAST homepage. This BLAST or the basic local alignment search tool finds regions of local similarity between sequences. The sequence may be DNA sequence, or the sequence may be protein sequence or RNA sequence. The program actually compares nucleotide or protein sequence to sequence database and calculate the statistical significance of matches. BLAST can be used to see functional and evolutionary relationship between sequences, as well as to help identify numbers of gene families. BLAST is more time efficient than FASTA by searching only for the more significant patterns in the sequences. Therefore, BLAST is more than a tool to view sequence aligned with each other or to calculate percent homology, but a program to locate regions of sequence similarity with a view to comparing structure and functions. So when you go to BLAST homepage, you will see the page like this, having different BLAST program. The BLAST program was designed at the NIH, that is National Institute of Health, and it was published in the Journal of Molecular Biology in 1990. As I told you in the previous slide, there are different types of BLAST program. Means based on your query sequence, it may be a protein sequence, it may be a nucleotide sequence. Based on that, there are different BLAST program. So if you are putting nucleotide query, you are having nucleotide BLAST. If you are putting protein query, you are having protein BLAST. Like this, we are having BLAST X, we are having T BLAST X, we are having T BLAST N. So when we do nucleotide BLAST, it will search a nucleotide database. When we do protein BLAST, it will search protein database. When we do BLAST X, it will search protein database using a translated nucleotide query. When we do T BLAST N, it will search translated nucleotide database using a protein query. When we do T BLAST X, it will search translated nucleotide database using a translated nucleotide query. So based on the query sequence, the BLAST program we have to select. Here in your practical, we will do nucleotide BLAST and protein BLAST. The steps are same, only the difference is your sequence. So here, if you are doing nucleotide BLAST, you have to put nucleotide sequence. If you are doing protein BLAST, you have to put protein sequence. In this particular practical, we will perform nucleotide BLAST. So we will need a nucleotide sequence. Here we will see how to input your query sequence. So as we are doing nucleotide BLAST, we have to select nucleotide BLAST from the BLAST homepage. Okay, if you are doing protein BLAST, you have to select protein BLAST from the BLAST homepage. So okay, so when you select nucleotide BLAST, again one form will come. So this particular form, they are having some particular points. and you have to follow them. The first thing is that you have to input your query sequence. As we are doing nucleotide BLAST, we have to input our nucleotide sequence. The sequence you directly input it, or you can input the gene bank ID number. Here we will just input the nucleotide raw sequence directly to the empty box which is marked in red color. Then there is one option called as run BLAST. Here you have to click, then the program will run and it will search the nucleotide sequence against the nucleotide database and it will give you result. Here I am showing you a nucleotide sequence which is used in this particular practical. As I am doing nucleotide BLAST, I am taking the nucleotide sequence from NCBI. If you are going for protein BLAST, you have just to download the protein sequence for a particular organism. Then you have to input this particular sequence in the appropriate box. Then you run the program, and in the next slide you can see the result. So here you can see the result for the query sequence. The sequence which is having more score with more percentage of identity is more similar to the query sequence. And one more thing we have to see, there should be less gap in the sequence and the query cover should be more. Then only we can conclude that the sequences are similar to the query sequence. If you want to know more about BLAST program, just go through this link. You will get lots of information. Practice it. Thank you.